pleasant day to all of us, especially to you, ma'am. Have a good day, and to my classmates also. Well, this time, I am going to share to you my ideas about gene in population, of how genetic transmission and the evolution of genetics in a certain population, and where each member of the population received its alleles from other member of the gene pool or from their parents, and pass it to the other member of the gene pool or to their offspring. Well, to understand how population genetics came into being, let us first have a short review about Gregor Mendel's and Charles Darwin theory. In 1589, um, Darwin's Origin of Species was published, in where he propounded two main theses. First is that modern species were descended from the common ancestors, and secondly is that natural selection was the major mechanism of evolutionary change. Well, only the first thesis won the acceptance of the scientific community. Uh, Darwin was deeply troubled by not having a proper understanding of the inheritance mechanism. It is maybe he had na siya'y kulang sa iyang study, either lack of evidences or proven nga mao gini siya ang kamatuoran nga ang evolutionary change is nagikan gyud sa natural selection. Well, so the assumption of Gregor Mendel's theory of inheritance was straightforward. It is the process of the offspring inherit discrete hereditary particles or the genes from their parents, which means the sexual reproduction does not diminish the heritable variation present in the population. But even after the rediscovery of Mendel's work, turned in 20th century, it was widely believed that Darwinism, evolution, and Mendelian inheritance were incompatible. So that is why kanang sila ang nag-open sa gene population pero dili sila ang naghuman. Either na ay mga changes sa mga study nila, but it doesn't mean nga giwala na ang ilahang mga theory. They are the first who discovered the genetic population or the population genetics. So the synthesis of Darwinism started the mark of the modern population genetics was achieved by a long and tortuous route. So let's continue. Well, population genetics is a subfield of genetics that deals with the genetic differences within and between the population, and involves in examination and modeling changes of frequency of genes and alleles in the population over space and time. It concerns with the genetic differences within and across the population. Mga genetic differences sa isa ka individual to in a certain population, kung unsay kalahian niya o asa siya na-belong, like that. Because when we say population, it refers to the group of organisms of the same species that are found in the same area and interbreed. So, population genetics, which help to explain the evolution of the population over time by combining the principle of Mendel and Darwin's theory by the means of Hardy and Weinberg equation. Through the theory of Mendelian and Darwin, si Godfrey Hardy and Wilhelm Weinberg nag-create sila og mathematical description of Mendelian inheritance. Well, Hardy and Weinberg equation shows that the frequency which you expect to find different alleles in a hypothetical population does not involve. In 1908, Godfrey Hardy, an English mathematician, and Wilhelm Weinberg, a German physician, independently discovered that an equilibrium is established between the frequencies of alleles in a random mating population. So, the two scientists who independently at the same time came up with the exact same equation that describes how, under the right circumstances, Mendelian genetics works at the scale of a whole population. So there are two equations in gene frequency and equilibrium. To come up with the idea of how gene frequency work, let us first study the formula of how P plus Q is turns to 1, where P represents the dominant allele or the big letter A, and Q represent the recessive allele or small letter A. This equation used to calculate allele frequency equal or 100% of the population. It doesn't mean nga A lang ang letter nga naa diha. Example na siya, either mo gamit ka small letter B, which makes your recessive allele. Second equation in where P squared plus 2PQ plus Q squared is equals to 1. In where P squared represents homogeneous dominant genotype. In where for example is homogeneous dominant 2 big W, it represents as P squared, while the 2PQ represent the heterogeneous genotypes and Q2 represents the homogeneous recessive genotype. This equation used to calculate genotype frequency equal to 1 or 100% in the population. Gihimo siyang 2PQ kay dili man siya mahimong dili po siya pwede nga mahimo siyang P squared kay lahi-lahi man sila lahi-lahi man sila ay ang isa is big W while ang isa is small w or small letter A and big letter A. So dili siya, it's either gibutang lang siya 2PQ. Next is, let's talk about gene frequency. So, as defined, gene frequency is a ratio of a number of a specified alleles in a population to a total of all alleles in genetic locus. Well, genetic locus means kanang kung aha na butang ang usa ka genetics is a specific fixed position. Asa nga position on a chromosomes where particular genes or genetic marker is located. So, gene frequency next is, in gene frequencies and equilibrium, na mga assumptions si Hardy and Weinberg na sila mga model. First is organisms is diploid. Blood diploid means only two alleles. Like as what stated here is that diploid is described as cells that contain 2 copies of each chromosomes. Production is sexual. Sa ilang model, dili ka makakuan kung dili sexual sa ang ilang production. Third is generation are non-overlapping. Mating occurs at random or random mating. Population size is very large. It is needed that to come up with this gene frequency, dapat ang population niya is very large. Migration is 0, mutation is 0, and natural selection does not affect the gene in question. Next is, since we know that homogeneous recessive traits has 2 recessive alleles, we can take the square root of the phenotypic frequency or big letter B and get small letter b that can be found as 1 minus b or is equals to a. So, kani nga example is dili man kayo maklaro sa inyo let's have another example about pea plant. So as we look in the picture, there are nine pea plants with different alleles. There are homogeneous, there are also heterozygous, and also the homogeneous dominant and homogeneous recessive. We can divide the number of copies of each alleles by the total number of copies to get the allele frequency. By convention, when there are 2 alleles of a gene in a population, their frequency are given the symbol P and Q. So, makapangutana ka nganong 18 man. In word, there are 9 only. Because they count nila as individual alleles ang usa ka flower. Like a flower is adunay 2 pair of alleles in which homogeneous and heterozygous. So, ilan ang gi-count as 1. So, 2, 4, 6, 8, 10, 12, 14, 16, 18. So, P or the frequency of W has a 13. In the illustration at the screen, there are 13 dominant, and also for the recessive or the symbol of Q, the frequency is 5 over 18, or there are 0.72 or 72% have a dominant, and there are only 28% is the recessive. So let's move on to the next. First is we're going to calculate the genotype frequency. Well, genotype frequency is how often we see each allele combination, such as WW, W small w, and small w. Allele frequency is different from genotype frequency or phenotype frequency. Genotype frequency and phenotype frequency can also be calculated or important for understanding how population evolve, but they are not the same thing as allele frequency, such as this. So the frequency of WW, homogeneous dominant genotype, is 6/9. So there are 6 homogeneous dominant genotype, and there are also one heterozygous genotype, and also the homozygous recessive of 2. So i-divide mo sila og 9. So 6 divided by 9, 1 divided by 9, and 2 divided by 9. So manana ilang mga answer, 0.62, 0.11, or and 0.22. Makita nimo nga correct yung answer if imuha na silang i-add, then the answer is equal to 1 or 100%. Next is, we will going to solve for the phenotype frequency, or how often we see white versus purple. In the phenotype frequency, there are only 7 purple pea plants. 7, yes. Divided by 9 is equals to 0.78, and the frequency of white is there are only 2 or 0.22. So after that, we were going to find the allele frequency. Well, the allele frequency is how often we see each allele. So the frequency of big W and the frequency of small w is 13. The big W is 13/18 is equals to 0.72, and the frequency of W is 0.28. So for the gene equilibrium, it is the condition of the allele or the genotypic in a gene pool, such as the population, in where the frequency does not change from generation to generation. Genetic equilibrium discussed as the theoretical state that is the basis for determining in what ways population may deviate from it. Example is the the heterozygous genotype, in where big letter A is represents as brown eyes. The phenotype is the brown eyes. The small letter A is the blue eyes. The dominant brown alleles mask the recessive blue alleles, or the genetic equilibrium is the state in which allele frequencies remain constant. In the gene frequency and equilibrium, gi-promote nila nga ang usa ka frequency sa usa ka allele is remain constant over generation to generation. Gene frequency and equilibrium. The Hardy-Weinberg principle states that for genetic equilibrium to maintain from one generation to the next, 5 conditions must be met. So, the 5 conditions must met that random mating. In random mating, all individuals in a population have an equal opportunity to pass their alleles to their offspring. And also, it must have a large population, because genetic drift or random change in allele frequency has less effect on large population than small population. And of course, there must be no mutation happen, because if genes randomly mutate, new alleles may be introduced into the population, and alleles frequency will change. And of course, no migration. That will going to happen, because either individual cannot move into or out into the of the population, because either kung may migrate sila in or out, either na masagol or makuha sa usa ka allele frequency. Manay promote nila mahimong constant ang ilang study if there is only a random mating, a large population, but no mutation, no migration, and of course no natural selection, in which all genotypes must have an equal probabilities of reproduction. So now let's talk about gene pool. As we mentioned earlier about gene pool, a gene pool is the total genetic diversity found within a population or species. A large gene pool has extensive genetic diversity and is better able to withstand challenges posed by the environmental stresses, such as the illustration below. Una siya ang mga kinds. Kinds of dogs under gene pool. And of course, as well as for the frog, na ay klase-klase nga frog, na ay green, na red, violet. So, mo na ilang gene pool, mo na nag-represent nila o ang total genetic diversity of a certain population. So, gene pool, the necessity of hereditary variation for evolution change to occur can be understood in terms of gene pool. So, assume, for instance, a population in which there is no variation at the gene locus that codes for MN blood groups. Only the M alleles exist in all individuals. Evolution of MN blood group cannot take place in such population, since the allelic frequency have no opportunity to change from generation to generation unless ma-change na siya kung na ay environmental changes or mga factors na maka-affect sa lahat. The term typically is used in reference to a population made up of individuals of the same species and include all genes and combination of genes in the population. The composition of the population gene pool can change over time through evolution. This can occur by a variety of mechanisms, including mutation, natural selection, and genetic drift. The result of gene pool is altered to be attuned to the needs of the population's specific environment. For example, the migration of human population from equatorial regions towards northern climate, where they were exposed to relatively low amount of sunlight, result in changes over time in the skin pigmentation, with skin becoming lighter in color to augment vitamin D absorption. The genetic modification underlying the change in pigmentation ultimately became a part of many other population gene pools. So now let's talk about Hardy and Weinberg law. Hardy and Weinberg law violation of assumption can cause deviation from expectation, and how this affects the population depends on the assumptions that are violated. If a population violates one of the following four assumptions, the population may continue to have Hardy and Weinberg proportion each generation, but allele frequency will change over time. So, in selection in general, causes allele frequency to change often quite rapidly, while directional selection eventually leads to loss of all alleles except the favored one, unless one allele is dominant, in which recessive alleles can survive at low frequencies. Some forms of selection, such as balancing selection, lead to equilibrium without loss of alleles. We have also the mutation. If we have a very subtle effect on the allele frequency, and the change in allele frequency will be at most the same order, recurrent mutation will maintain alleles in the population even if there is a strong against them. Migration, also genetically linked to two or more populations together. In general, allele frequency will become more homogeneous among the population. For those models, Hardy and Weinberg proportion were normally not valid. For example, in a certain population, either ni-migrate sila pagawa sa usa ka population, in which the allele frequency is nagkagamay pod. Ang klase-klase nga alleles nila is nagkagamay ang probability. Ang probability in which nga na possibility nga mahimong homogeneous ang usa ka population. And also, small population size can cause random change in allele frequency. This is due to the sampling effect and is called genetic drift. And genetic drift later on i-discuss na sa next reporter kung sige ang meaning sa genetic drift and why did it happen. Always remember that in population genetics, Hardy and Weinberg principle states that alleles and genotype frequency in the population will remain constant from generation to generation in the absence of other evolutionary influences. Always remember that there are 5 assumptions of Hardy and Weinberg principles. First is the random mating, all large population size, no mutation, no migration, and no natural selection. But if this assumption will not met, then there will be a changes of genotypic frequency or the allele. So that's all, and I hope you got some idea about population genetics that I have discussed to you and how we solve the genetic frequency and the equilibrium. That's all, and thank you.